Hello, I'm Jose Antonio Sanchez Alcazar from Pablo de Olavide University. Today I'm going to talk to you about a subtype of neurodegeneration with brain iron accumulation named phospholipase A2G6 associated neurodegeneration, or PILAN, ultra-rare genetic disease. Neurodegeneration with brain iron accumulation is a group of inherited neurological disorders in which iron accumulates in the brain. Pathophysiologically, many of the NBIA syndromes map into related biochemical pathways and gene networks, including iron and lipid metabolism. At least 15 genes are associated with the different types of NBIA. In this talk, I will focus on PL2G6 mutation, which is a particular subtype of these disorders. PILAN is an autosomal recessive inheritance disease with a prevalence of 1 in 1 million, and it presents 3 disease phenotypes with different onset age. Infantile neuronal dystrophy with an early presentation before the first 2 years of life, atypical neuronal dystrophy with presentation in the early youth, and PL2G6 related dystonia Parkinsonism, which has a late onset in adulthood. Infantile neuronal dystrophy, the most studied subtype, usually begins between age 6 months and 3 years with psychomotor regression or delay, hypotonia, and progressive spastic tetraparesis. Strabismus, nystagmus, and optic atrophy are common. Disease progression is rapid, resulting in severe spasticity, progressive cognitive decline, and visual impairment. Many affected giants do not survive beyond their first decade. In PILAN, MRI images shows diffuse symmetrical atrophy of bilateral cerebral hemisphere. In addition, images shows hypotensity in symmetrical manner involving bilateral globus pallidi and substantia nigra, suggesting mineral deposition. The disease is caused by mutation in the PL2 gene encoding an enzyme which catalyzes the release of fatty acids in SN2 position of cell membrane. This enzyme has a central role in membrane remodeling by removing peroxide fatty acids from membranes. In addition, it is involved in cellular signaling because hydrolyzed phospholipids release arachidonic acid and lysophospholipids, which act as signaling molecules. The deficient activity of PL2G6 affects all cellular membranes, including mitochondrial membranes, and therefore affects the proper mitochondrial function and the generation of energy. Increased lipid peroxidation also leads to the generation of various reactive aldehydes, and these aldehydes trigger several redox signaling pathways and cellular damages, including alteration of transcription factors, autophagy, and mitochondrial function. In this work, we have studied 2 patients with PL2G6 mutation, one with infantile neuronal dystrophy and other one with atypical neuronal dystrophy. As biological model, we used patient-derived fibroblasts, which allow us to do personalized medicine. First, in the mutant cells, we examine the main pathological alteration associated with the disease, such as lipid peroxidation and iron accumulation. Initially, we analyzed the expression levels of the mutant enzyme in control and PILAN fibroblast cell lines, and results showed that PILAN fibroblasts presented reduced protein levels of PL2G6 in comparison with controls. As expected, mutant cell showed increased lipid peroxidation in cell membranes assessed by body P staining. Pilan fibroblasts also exhibit an increase in iron staining by Prussian blue compared to controls. Iron accumulation in Pilan cell was also confirmed by colorimetric ferrocene-based assay. Interestingly, Pilan fibroblasts presented an enlarged and flattened cell morphology similar to senescent cells. As lipofuscin accumulation can result from lipid peroxidation, a process also stimulated by iron, we next examine the presence of lipofuscin by autofluorescence analysis, Sudan black staining, and electron microscopy. Here we can clearly see the increase in autofluorescence and Sudan black staining in Pilan fibroblasts. And in electron microscopy images, we clearly observe the presence of lipofuscin granules. Next, with the aim of knowing the relationship between lipid peroxidation and iron accumulation, we treated healthy fibroblasts with luperox, a lipid peroxidation inducer, and with iron to induce iron overload. As shown, peroxidation induced by luperox led to increased iron accumulation, and vice versa. Iron overload induced lipid peroxidation. Furthermore, we also assess the presence of lipofuscin granules under luperox-induced lipid peroxidation and iron overload conditions. Both luperox-induced peroxidation and iron overload induce an increase of lipofuscin accumulation. These results suggest that both processes, lipid peroxidation and iron accumulation, are interconnected and they reinforce each other in a kind of vicious cycle. Next, we evaluate the effect of vitamin E treatment, a well-known membrane antioxidant, on lipid peroxidation. As expected, lipid peroxidation in Pilan fibroblasts was significantly reduced under vitamin E treatment. We also evaluated the effect of vitamin E treatment on iron accumulation. Vitamin E supplementation reduced significantly iron overload in Pilan fibroblasts assessed by Prussian blue staining and reverted their senescent morphology. Reduction of iron levels by vitamin E treatment was also confirmed by inductively coupled plasma mass spectrometry and ferrocene assay. Vitamin E supplementation also induced significant reduction in Sudan black staining and autofluorescence intensity in mutant Pilan fibroblasts, suggesting lipofuscin elimination. Furthermore, vitamin E treatment prevented superoxide accumulation in Pilan cells and also improved Pilan cell viability in the presence of luperox, an inducer of lipid peroxidation. Specifically, high levels of lipid peroxidation in mitochondrial membranes were present in Pilan fibroblasts. As expected, vitamin E treatment significantly reduced mitochondrial lipid peroxidation. Pilan patient-derived fibroblasts also showed a reduction in mitochondrial membrane potential associated with increased mitochondrial network fragmentation. Interestingly, vitamin E treatment improved both mitochondrial membrane potential and mitochondrial network. Next, patient Pilan fibroblasts and control fibroblasts were transdifferentiated into induced neurons by transducing lentiviral vectors containing proneural genes to allow the investigation in one of the most affected cells in Pilan disease. Here you can see a monolayer of culture fibroblasts, which are efficiently transdifferentiated to induce neurons by direct reprogramming. 27 days post-infection, cells presented neuron-like morphology and positive immunoreactivity against tau. We use tau cells to evaluate neuronal conversion efficiency. Neuronal electrophysiological properties were detected in generated induced neurons. The resting membrane potential of neuron-like cells were around -24 millivolts. Induced neurons also show an increase in iron staining and superoxide production compared to controls. Furthermore, induced neurons also presented a higher area and perimeter of neuronal soma than control-induced neurons. As expected, induced neurons showed an increase in peroxide lipids compared to control, and furthermore, vitamin E treatment of induced neurons induced a significant reduction in lipid peroxidation. Vitamin E treatment in cells induced neurons also show a positive effect of attenuating iron accumulation and reducing significantly neuronal soma area and perimeter. Vitamin E treatment also reduced superoxide production in induced neurons in comparison with untreated induced neurons. Finally, vitamin E treatment significantly improved mitochondrial membrane potential of induced neurons. In conclusion, patient-derived fibroblasts are a promising model for the study of disease pathophysiology and the evaluation of potential therapies. Fibroblasts are induced neurons harboring PAL2G6 mutation, manifest, assess lipid peroxidation and RAX production, iron lipofuscin accumulation, and impair mitochondrial membrane potential. Induced neurons can reproduce the main pathological alteration of the disease and are also a useful tool for disease modeling. The role of lipid peroxidation in iron and lipofuscin accumulation suggests that this process is a main therapeutic target. To this respect, we show the protective effect of vitamin E supplementation on the main pathological alteration in both cellular models. These findings suggest that blockage of lipid peroxidation by vitamin E supplementation can be beneficial in patients. Another important conclusion of our finding is that lipid peroxidation and iron accumulation are interconnected processes that reinforce each other and may aggravate and accelerate the progression of neurodegenerative diseases. Our work is a good example of how the investigation in an ultra-rare genetic disease such as can provide key information about the pathomechanisms of more frequent neurodegenerative pathologies. The findings of this work can be relevant for many neurodegenerative diseases associated with brain iron accumulation, such as PCAM, BPAM, MPAM, FAM, Alzheimer's disease, Parkinson's disease, Huntington disease, amyotrophic lateral sclerosis, Friedreich's ataxia, etc. Finally, my gratitude to my collaborators who have made all this work possible and patient associations which support our research. Thank you very much for your attention.